Hi all, we are continuing with viruses today, and the goal really is to talk about how viruses have a role in controlling, we'll say, populations of different animals, plants, and humans. Um, how viruses are pathogenic in nature. We're going to talk about how we can protect ourselves with vaccines, and you're going to watch a video on how this vaccination process works. We'll then review your notes and we will assign you a bit of work. So, what I would like you to do before you start this lesson is pause the video and just write down 5 or 6 different things that you remember about viruses. The kind of things that you should be mentioning here would be, for example, viruses are parasites. Okay, that means that they will invade and rely on hosts' cells to survive. So they cause, I suppose, harmful effects to other organisms and they benefit themselves. You might have mentioned that there are different examples. So we know, for example, HIV is something that affects humans. We know that viruses are smaller than cells, much smaller, in fact, that they are is really DNA and RNA surrounded by a capsid. You may have mentioned other things as well, like for example, viruses can be used to create vaccines, and you may have mentioned as well that viruses are, I suppose, many of them are pathogenic. They cause disease. So skipping on from that, what else do I need to know? Well, you need to know that they're what we call obligate parasites. And the way I like to think about this is that they are obliged to be parasites of cells. In other words, that they cannot survive without a cell. What we mean when we say obligate parasites, the definition, we say that viruses can only replicate within another living cell. So that means when we talk about replication, that they can make clones of each other. Remember, the goal of a virus is once it gets into your body, it needs to find a suitable cell to invade. It needs to take over the cell and it's going to use that cell to replicate. And when it replicates, you start off maybe with one virus. That virus becomes many, many viruses. In fact, when the cell has finished its life and it has been infected, it might release out millions of that virus and millions of copies of that virus. So um, we say they're obligate parasites because they can only replicate within our living cells. Right now, I suppose COVID is the virus we're trying to defeat. And so when our immune system isn't being activated, we're really trying to attack our own cells that have been infected with this virus. And that's why it's taken such a toll on our immune system. So you need to know different examples of viruses, and there's many different examples of virus. Some of them, I suppose, some of them we would consider fairly harmless, and others would be much more deadly. Like for example, herpes virus is something that can occur, causing cold sores, which in its own right, I suppose, is a bit harmless. But then we have human immunodeficiency virus, HIV, otherwise known as AIDS, which is another virus that affects humans and is deadly. Um, Ebola is a virus again that affects humans, and we've heard of the Ebola outbreak that which happened mainly in Africa. Um, we have other historical diseases, and one we'll be seeing in a video later, which is smallpox, which was highly infectious and dangerous back a few centuries ago. So those are all examples of human diseases caused as a result of viruses. If we're talking about animals, as I mentioned yesterday, we could be talking about things like rabies. And if we're talking about, for example, plants can be infected with viruses as well. For example, there's something called mosaic disease, which causes a discoloration of plants, and that discoloration, I suppose, is affects the ability of the plants to photosynthesize, etc. So that's called mosaic disease and affects tomato plants and tobacco plants. So what I want you to do is pause the video, and I want you to skip on here and have a look at this video which I have posted on the Google Classroom, and this is called The Origin of Vaccines. It'll talk you through how vaccines started, with a guy called Edward Jenner, and he started experimenting on cowpox and smallpox to try to develop something called a vaccine. So once you've watched that video, you can pop back onto this then, and we'll just talk in general about how this vaccination process works. So let's put this in context. Right now, we are trying to, I suppose, as a society, develop more and more vaccines for COVID. There's about 3 vaccines on the market right now, and how have they been produced? Well, what you do is, when you're trying to develop a vaccine, is you're going to try to go through animal trials and human trials. So, in order to develop a vaccine, you have to infect uh, first an animal or a human with the virus. Now, you're not giving them the disease causing version of the virus, you're giving them what we call a weakened or inactivated form of the virus. So, you're going to inject it into their bodies, will say, circulatory system, and you're going to see how the body responds and to see can it produce what we call antibodies. Now, what you're really introducing in, so as we know, when viruses enter the body, they enter cells. And when they enter cells, they leave behind what we call an antigen on the surface. And an antigen is just a protein that will actually be recognized by white blood cells. So all cells have antigens on the outside of them, but the viruses will leave behind a specific type of antigen which can be recognized by the white blood cells. So let's go back to what I was talking about. What has the doctor done in their trials? They may have introduced, we'll say, for a human trial, they may have injected a person's blood with the weakened version of the virus. When they introduce the weakened form of the virus, it may actually be that they have introduced this antigen that was left on the surface of the cell. So this is a viral antigen. When that was introduced into the body, it takes a while for the body to recognize that this is a foreign body, that this is something that's foreign, that shouldn't be inside in the body. So the white blood cells are just floating about until an alarm bells go off and they recognize that we have a foreign agent that's inside in the body. Immediately, with this, the white blood cells go out to investigate, and they try to find the infected cells in the body. Once they find the infected cells, they start producing antibodies. So antibodies will attack a cell that has been infected by the virus. But unfortunately, many of these antibodies will not work at first. It takes a while for the white blood cells to produce the correct antibody. So when they produce, when it might just be that one white blood cell in the body, after a period of days, will find the correct antibody, and then it will produce it will reproduce. The white blood cell will reproduce many times, and suddenly now the body is full of these white blood cells which have the correct antibody to kill the virus, and that memory will be stored in the body. Now, I'll be talking about this further when we look at the immune system in detail, but what do I want you to remember now? I want you to remember that when you vaccinate, when you're really introducing part of the virus into the body which has been weakened. So you've weakened the virus and you've injected it into the body. You're hoping that the body will recognize this as a foreign agent. So, you can imagine the spy movies or whatever, that they're recognizing their agents, and it then produces antibodies. It takes a while, but eventually it'll produce the right antibody. And if it produces the right antibody, then over time this antibody will be remembered in the immune system. So you go through animal trials first and see, if you introduce the virus, will the animal produce the antibody to kill the coronavirus? You then go on to human trials and see if it works in animals. We'll try it on humans, but animals and humans, they're not exactly the same, so it may or may not work. And if you go through enough human trials and you prove that for most people that this will give them the ability to fight the infection, then then you might have approved the vaccine and said, okay, this is ready to be used on a large percentage of the population as a vaccination. Okay, and we've been doing this for years, because we have been doing this for example, we've been doing this for measles, mumps, and rubella. Now, I don't think I have these listed off here, but what are the benefits, of course, of vaccines? The benefits are, well, one, you prevent a very harmful disease. So, viruses, if we said what is the role of viruses in nature, we could say part of its role is population control, because for large amounts of animals and plants and humans in history, it reduced down the population, which meant that there was less competition after disease had kind of ravaged the populations. So ecologically, viruses have a very important role. But for us as humans, what's our benefits of developing vaccines? We could say, well, it prevents these harmful diseases. We can also use them to vaccinate our animals against harmful diseases, so they're healthier. The disadvantage of viruses, of course, is that, well, they are there and they can cause harmful diseases, and they can cause sickness, or they can cause disability, and viruses can be very dangerous. Okay, so that is it. It's a short chapter, and I just want to review now the notes that we've taken down on this to make sure we're all happy. So we have talked here about all of these different things, like, for example, obligate parasites. They replicate within cells. One thing I mentioned at the start of this chapter was the fact that viruses will eventually cause the cell to burst, and we call that lysis. We talked about replication, that they copy DNA. So viruses undergo replication, where they copy their DNA or RNA. And just be aware as well that bacteria can be infected by viruses as well. These are the two diagrams that you should be able to draw when you're representing viruses. So this is a virus itself, and this is its DNA or RNA. It has a capsid that surrounds it. And the other thing then is a smaller diagram. So this could be another type of virus, and this is the host cell that it's infecting. Right. There's a few other benefits here that I mentioned down the bottom. So viruses, of course, used for vaccines. We also will be talking about them when we do genetic engineering, okay, because they're used in genetic engineering, and they're just generally used for medical research. Finally, then, you need to be able to talk about, I suppose, examples, as I said, and how replication works, and we discussed this in class yesterday. So once you have all these notes taken down, that's it. That's the exam focus. You have everything you need. Okay, so next chapter we'll be looking at is bacteria.